Michael, for the organization of this meeting. I hope you can now see the screen all. And I feel very honored that I'm the first actually of this series, but perhaps it also makes some sense because this very first lecture is really one on very profound mechanisms of neurobiology. And the aim of the work in my group is to unravel some of the cellular mechanisms that might lie behind cognitive or neural reserves. So, of course, as most of you are aware, people age differently, and while this phenomenon is well known, we know very little about the underlying mechanisms. We are pretty good by now to describing this phenomenon, and there are a lot of first steps to use it. But what really constitutes these reserve or maintenance at the level of the brain itself is not really understood. And I take this serious and the related research that people in this series do as an indication of the growing interest in such mechanisms. So we are looking at the half full glass rather than the half empty glass in neurodegeneration, and ask what is the brain set against the problems of cognitive aging and neurodegeneration. Now, that there is something in lifestyle and related aspects that is to some extent preventive from neurodegeneration is of course not new. And last year the WHO has issued, actually for the first time in its history, and doing something like that, a guideline for risk reduction in cognitive decline and dementia. And when we look at the key messages that are in this rather thick volume, then we find recommendations such as don't drink too much, don't drink too much alcohol actually, exercise regularly, don't smoke, manage your blood pressure, maintain a healthy weight, eat a balanced diet. And most people will have said, or actually did say, hey, wait a minute, WHO, we've heard that before. Isn't that the list of things that's good against anything, everything? Cancer keeps you, oh, this is, what's so specific here? And the truth is nothing, presumably. These are all very general recommendations, and they're good for us for various reasons. They keep us probably healthy if we obey to them. But what does that have to do with dementia? So doesn't it lead to some circular argument? If you're more healthy, your risk of dementia is decreased. Is there anything specific here? Ultimately, this is very difficult to disentangle. But the question of some specific mechanisms in the brain that might be distinct from others is actually quite urgent if we really want to understand this. And even more pressing is the question, is this all? I mean, if a recommendation makes it to the level of the WHO, it must be really based on rather solid evidence in many studies. But of course, this might not be all. So I've gone through the literature and collected information from studies that to some extent have dealt with the question, is there some, is such and such activity good for the brain and can be preventive from neurodegeneration? So the level of evidence is very different in these studies, but they're all scientific studies, have been published, they have gone through peer review, and they have some some evidence to them. And this number is much larger of these factors. And now we find things that are very different from the things that made it on the WHO list. Sleep is here, cognitive stimulation, education itself, all kinds of physical activity in more detail. Down at the bottom here, you see music, dance, all things. And you can arrange them in such scheme where you have these 4 quadrants, and doing so is actually a little bit telling, because it points to some interesting effects. So it would be a 4-field matrix with body and mind as the columns, and input and output as the rows, and we can sort our lifestyle factors according those lines. And that then it creates some fields of tension, or some contrast, like receiving versus giving, being influenced by versus influencing, activity versus passivity, object versus subject, and on the other axis, mental versus physical, self versus social, energy versus information, quantity versus quality, and so on. These are not strict dichotomies, obviously, but they create a picture that highlights that all these factors, which have some, at least face value, face validity, to be preventive or neuroprotective, are actually not uniform. Whereas this WHO list, and so they focus on just a particular type of thing. Now, why is it so difficult to adhere to these things, and why is the bias that these lists that are published of these high-level recommendations are not more effective? Well, perhaps it is because we assign different value to these things, and we more or less intuitively have this pyramid of values in our heads, which has a relatively broad physical basis, and then we stack emotion, mental, and finally the spiritual values on top. And if you look at this arrangement of the lifestyle factors, you see that there are actually lifestyle factors that we consider extremely important to our life in general, so more at the spiritual level. And they fall into this lower right quadrant, the purpose quadrant, which is not as easily tangible as the stuff that you find in the physical world of diet or exercise. Now that creates a discrepancy of what we measure is not the same in terms of these factors as what is actually done, because the paradigms, intervention paradigms, they fall into a different category. And there is a whole domain that we value very highly that is actually missing. Now, if we look at all this, we cannot really escape the notion that there must be something that is sort of in the middle, something that brings together this this field of tension, so that the sum of all this is, or that the entire concept is more than the sum of this individual identified interventions or parameters. So what is meant is at the end something that is in the center: quality of life, balance, homeostasis, and so on. You can happiness even. You can have various concepts in the middle that you would see. Well, this is the essence of a balanced life. Now, what does this have to do with mouse research as we do it? Obviously, it's very hard to look at happiness or such concepts in a mouse, or religious life, and there's no yoga studies in mice and so on. But still, what we can do in research, in pretty much every type of organism that we can study, is that we measure more than one thing. And that also here we move away from measuring only the things that are easy to measure and where we can get good numbers, but that we extend the scope of our research so that we get to a bigger, or a larger panel of parameters, make the research multivariate. Now, if we look into the history of research on brain plasticity in the sense of this more general brain plasticity, how does the brain respond as a whole to learning, to stimulation, we come to this large body of literature from so-called enriched environments, concept invented by Donald Hebb in 1949 and then brought to much prominence by for example, Marion Diamond, Mark Rosenzweig, Edward Bennett, David Craig in Berkeley, and many other researchers like Bill Greeno have contributed to that. And it has been an extremely influential paradigm, by and large always showing that when you expose research animals like rats and mice without any other intervention, just to a larger cage with more mice or rats in that case, you get profound and very very clear and replicable changes in the brain. Well, pretty much all parameters that you measure. And Marion Diamond has captured the essence of the concept of that time in the title of her book on the subject, which was Enriching Heredity. So the idea in that sense is that when we have this classical equation of the phenotype equals the genotype times or plus the environment, then the concept of environmental enrichment, the paradigm, is that you keep the genetics constant, you don't change that, and you would look at the effect of environment on the phenotype. So the paradigm then looks as such. You have a control condition, which is standard housed condition, and importantly, the animals are treated well. There's no, it's not a negative trim. It's just no additional beneficial treatment, and then you offer more animals, more space, more room, more change, more things, more stuff to explore, and that's called enrichment. So it's a kind of sensory multimodal stimulation. Now this is so you compare then effect between baseline and compare these two groups on these lines. We come back to that other aspect in a moment. So when we look at parameters in such animals, this is a couple of behavioral data. Then there is something intriguing. Well, first of all, we can always see that here you have in the greenish yellow, you have the enriched group. If you compare those with the controls, you have a mean effect. Here on spatial exploration, on the habituation, on the open field, on the novel object recognition task. You don't have to go into the details of these paradigms, but we see differences in the mean. So there's group differences for example, for these behaviors, simply by exposure to the enriched environment. Nothing else has been done. But now there's an additional observation which hasn't received much attention. There's actually very few mentioning in the literature of this field, which is amazing because it's over, since 1949 we have papers on this subject, and that is that when you look for these parameters on the enrichment in the enriched animals, you also see that the variance of the values increases. So it's not just the mean, but also the variance. Now there could of course be the fact, because the mean changes, there's also a correlated effect on variance, but this is not the case. This effect on the variance goes beyond this effect, and you can calculate this statistically with a Brown-Forsyth test, for example, and then you find that there is an enrichment effect on variance, not just on the mean. this also applies, interesting now, to morphological parameters in the hippocampus as a main structure, brain structure for learning and memory, and of course prominently affected in Alzheimer's disease and other dementias. And this doesn't apply to all other brain areas. So hippocampus is kind of special here when we look at N here, for example, the cingulate cortex, we don't see that effect. Entorhinal cortex, we don't see that. But we find a difference at the level of the motor cortex, for example. So here we find a mean effect, but there is an intriguing effect on one particular brain parameter we'll come back to in a moment. But the point is, we have effects on variance, we have effects on mean, we have, they're not always correlated, we have these sometimes on structure, we find this link, sometimes we don't. And the observation of an effect on variance, of course, can be taken as some indication of individualization in this condition, because if the genetics are the same and the environment is pretty much the same, then we have to ask, where is this differences? What is the source of this differences? Now, this is actually a very interesting question and might be very relevant. And surprisingly, it has been asked so rarely. Most of the time, actually, people argue that enrichment does not affect variance. And a more recent paper on this is a study here from Munich, from the mouse clinic, where they looked at a large number of mice, even from 2 strains. And there's here a variance indication, and they say there is no change in variance, but then they report here in this figure 5 that there's actually quite substantial effects in variance. So the colored dots indicate that there is some effect on variation in these groups indicated by the columns, and for all these parameters that the mouse clinic can assess. It's relatively small effects, but it's also relatively minor intervention that they do. The enrichment consists essentially of a little plastic shelter in the cage. So I thought this despite the tenor of that paper, it's actually quite interesting that they also seem to find that when you enrich animals, you see, compared to a control condition, an increase not just in mean effects, but also in variance. And this is not consistent or uniform across the panel of parameters that is studied. Now, we are particularly interested in one, I told you that the hippocampus is somewhat particular here, and we are interested in one particular subphenotype in the hippocampus, and this is adult neurogenesis. In adult neurogenesis, we have a exception to the otherwise iron rule of the brain that it doesn't produce new neurons, and that's the mossy fiber connection between the dentate gyrus and area CA3 is subject to the generation of new neurons pretty much throughout life. And my personal research interest is in the activity-dependent regulation of this process and the question how it might contribute to a lifelong shaping of the hippocampus to meet the challenges that an individual has in life, throughout life. Now, this is a trait, a plasticity trait, and unlike synaptic plasticity, which is of course found everywhere in the brain, this is really happening at the level of cells. So you can, it's a rather large unit in this network, and the analysis is much more straightforward if we look at entire cells rather than trying to capture what's going on at one of the myriad of individual synapses. So neurogenesis is a particular plasticity phenotype, but it happens in a brain structure important for learning and memory, very relevant for Alzheimer's disease, and it is a phenotype that is strongly influenced by behavioral activity. So when we look in this enriched environment situation within the hippocampus, specifically at adult neurogenesis, then we can see that, and this is the middle two panels, that the production of new neurons in this is not just massively enhanced in the control, in the enriched versus the control group, but there's also this described effect on variance. That is, the plasticity, the cellular plasticity in the hippocampus is affected by the environmental stimulus, but not just as a group effect, but within the group of the enriched animals, there's also huge variation. You can already see in this panel with the new neurons that there are some animals in the enriched group which do not really differ from the controls at all, whereas others have, in this case, almost fourfold as many neurons. So our idea is, in general, that adult neurogenesis is a trait that allows to adapt hippocampal function, which in turn improves or at least changes the behavior of the animals in their environment. And this by itself, this behavior of the individual has a feedback on neurogenesis, but it also changes, of course, the experience that the individual can make. And this again has an effect on neurogenesis. So it's a twofold feedback, positive feedback loop on plasticity. Now, as I said, there's relatively little, has been relatively little interest in many, for many decades in the neurobiology of this individuality or individualization. One of the papers, of the old papers that actually mention this point and make an argument is this paper here by Arthur Wimby and Victor Dannenberg, and from 1966, and they come here in their story to the hypothesis, one should say, that the results establish that stable and relatively permanent complex individual differences of the sort often assumed to be genetically determined can be generated by the appropriate manipulation of experiences during early life. So an idea of creating or stimulating individuality by exposure to stimuli. And this is a different way than looking at this enriched environment concept. Because if we really look at this classical paradigm, we realize that we are not actually manipulating the environment when we look at this group, because the environment is normally also the same. There's a comparison between control and the group, but otherwise there's no real manipulation of the environment. The only ongoing change in stimulation is actually happening by the animals that live in that environment, and the experience of this change, and this is called the non-shared environment. It's the aspect of a set of stimuli that has a tendency of making us different rather than making us more similar. So the idea is that this enriched environment may actually be a model to look at the non-shared environment and the effects of non-shared environment. As we have already seen, it seems that not all parameters are affected by this, but that you can see such effects across quite a range of parameters. And adult neurogenesis as a hippocampal parameter related to a concrete feedback loop, this effect is actually very strong. So we can reinvent sort of the enriched environment paradigm, and not just using it in the classical sense of comparing cross-sectionally between the groups, but also longitudinal within animals, or within, and at the same time cross-sectionally within one group, within the enrichment group. So to look at variation, to look at variation over time. So you can make it longitudinal, you can make it We can make it longitudinal, we can make it looking at the individuality within the group, and at the same time we can also make it multivariate, so looking at more parameters. And with that, then we have a new tool for looking at complex interactions between lifestyle, so to speak, which is of course a slightly different concept in the mouse than in humans, and brain plasticity over time. So we also needed to build a new gauge for our mice that would allow to test this better. And this is sort of the Buckingham Palace style of enriched environments. You know, Buckingham Palace has 700 rooms, and we use 70 rooms for our mice. And it's normal cages that are connected, so the mice can roam through all these different cages. And we monitor their activity, and we monitor their movements throughout this large area. We do nothing else to them. You know, just have them in the cage. They just live there. They have toys and plays like in a classically enriched environment, but there's nothing else. No intervention. And when we look at the activity, you can see at the bottom that the mean activity over time slightly decreases. Animals get habituated to that. It's not that exciting anymore after a time, perhaps, but still, activity remains fairly high, but there is some decrease. But variance goes up. That means that the longer these mice live in that cage, the more different they become in terms of their activity within them. And again, nothing is done to them. It's just the exposure to this cage. They're genetically identical. The environment is identical, but still they become more different. And we know from these other studies already that there are changes in their brain as well. So what's the relationship between such changes that we see them in these classical studies on variation now to these trajectories? We published on this already quite some time ago, that these diverging behavioral trajectories indicate relatively stable, or that these diverging behavioral trajectories are relatively stable over time, and that at the end we correlate them with adult neurogenesis. We can see that roughly 22% of the variation, so 1/5 of the variation in adult neurogenesis, can be explained by these different behavioral trajectories. Yeah, so we have a concrete relationship between a longitudinal behavior, a stable behavior, and a brain plasticity measure. And that is a quite fundamental difference now that allows us to really focus on change based on individual activity, because we have a parameter that is very sensitive to this, adult neurogenesis, and also, by the way, the novel object recognition task that I mentioned in the beginning. And we do not have to make any assumptions about the type of intervention that is needed. We can leave that open. It's still not the full circle of things that we see from the human studies and the literature that I shown to you in the beginning, but we can leave it open. We just leave it to behavior and can ask what are the sources of variation in such a context. Yeah? And then, of course, relates to this idea of Robert Plum and Denise Daniels' famous paper from '87 on why are children of the same family so different from one another, so that siblings can be more different than randomly picked children from the population, despite they sharing so much genetic identity. The idea here from these papers is this concept of the non-shared environment, and so that a very substantial part of the variation that we can see is due to this non-shared environment. And the non-shared environment, the behavior of the individual in this, of course, has an impact on the shared environment, because the environment might actually change by the behavior of the individual. So it loosens this whole concept a tiny bit. Now, if we go back to the question of the mechanisms, now we have this paradigm of the adult neurogenesis as a exemplary parameter, which is interesting. Then or that we can study in this model, then we still have to ask what constitutes a mechanism here. And this is one of the big challenges here. Very difficult to answer, actually. Quite philosophical. What is a mechanism? What would we explain? What would we accept as a cause in such a situation? It's really like a nest. The mechanisms in this sense are really nested from the genetic makeup, genetic background to the organism, and you can extend that even beyond to culture, social structures, and so on. And the effects of environment have to penetrate all these different layers to go down to ultimately to the DNA level, because there is genetic regulator, gene expression is at the heart of change. You will not get plasticity in the brain if there is not at some level a change in gene expression in some cells, and that leads to these alterations. And vice versa, you have an outward, outbound direction here that goes back from the DNA all the way to the rest, so that there must be consequences of change at the genetic level that ultimately lead to change in behavior and even society. Yeah, you can depict that in a slightly different way here, and contrast this inward causality of the world and how an individual reacts or lives in its behavior with an outward causality, which goes from the genetic makeup and other intrinsic determinants to the social interaction and behavior. And this outbound and inbound causality traits, they of course meet somewhere. In some sense, they meet everywhere, but perhaps a phenotype or a trait like adult neurogenesis, as we look at, is very well suited, somewhere in the middle, at the level of cells and a distinct circuit, to look at these two directions of causality. With that, I would like to summarize for you what I've tried to tell you today. A refined version of enriched environment paradigm is an experimental paradigm that allows us to study in a highly reductionistic way, admittedly, the neurobiology of individual risk and resilience. Adult hippocampal neurogenesis is a very concrete example of such a neurobiological reserve mechanism. We don't know how relevant that is for the big picture in humans, of course, but it is a mechanism that we can study to understand principles for even more elusive traits, perhaps. And longitudinal multivariate implementations of enriched environments can expose the non-shared environment in our experimental settings, even when we keep the genes and the environment constrained. And by extension, adult neurogenesis is actually an individualizing trait, so that we become more different because our actions make our hippocampus different. With that, we learn differently. With that, we might age differently. Thank you all for listening today. These are, whoops, this was not meant to be. These are people who did the work, the studies that are presented to you today by Jadna Bogado-Lopez, Anna Gip, Rupert Overall, Annette Rünker, Susan Schilling, and Sarah Zocher. Other people in my lab supported us. The studies relied, of course, on our funders. They were great collaborators, Ulman Lindenberger, Antonio Krüger, and others who helped us establish this, this research. Again, thank you for your attention today, and I'm happy to take some questions.
Yeah, thank you very much, Gerd. This was a fascinating talk. So I would like to ask you now to pose any questions. So now's the opportunity. Please don't be hesitant. This is really meant as a chance for you to interact and connect. So please raise a hand and I will bring you online. So I'm not seeing anybody yet. So maybe, Gerd, I'm starting with a short question. So it's really.
Sorry, Michael, Ian Robertson here in Trinity.
Okay,
I don't know how. I don't know how to raise my hand, so I don't know how to do that.
Yeah, please go ahead.
Thank you very much. A fantastic talk, Gerd. Thank you very much. Just, I may have missed this, but this fascinating phenomenon of increased behavioral variance, even with no intervention, and also increased variance in neurogenesis, and the two being correlated. Have you any idea about cause and effect? Does the increased behavioral variance precede the increased neurogenesis, or vice versa, or is there some third mechanism causing the correlation?
Yeah, that's a great question. It's really the heart of the question that you are trying to address. Is this a unidirectional causality? So is it coming from the neurons? We know that there's change in neurogenesis, and we think that, as I proposed, that it's a feedback loop, so that one is causing the other. We have, of course, ongoing experiments that manipulate neurogenesis and look at the effects on individuality. This, of course, will be very interesting. And one can, of course, also start trying to manipulate the environmental experience and make it more hippocampus, more stimulating to the hippocampus, for example, and see whether that accelerates the effect or changes this relationship. So I think at the end, it will be all of the mentioned, all of the points that you mentioned. There will be shared causality of some things that we just don't know. This would be at the heart of this idea that that this is highly multifactorial, but that it's a very good readout, but also that it's not just an epiphenomenon, but because it involves the hippocampus, it's actually also causally involved in this changed behavior, and by extension then with the individualization of the hippocampus.
May I, sorry, Michael, may I have a follow-on question?
Sure.
So Gerd, there was a lab in Lyon a few years ago studied, it was the olfactory bulb, neurogenesis in the olfactory bulb. So it's all smell behavior in mice. And they looked at, they showed that noradrenaline was the key mediator between the enriched experience and the neurogenesis in the olfactory bulb. And they showed that novelty was the key, if you like, behavioral dimension of the enriched environment rather than complexity. Any thoughts on that?
Yeah, there's quite a body of literature in some of them very old, on these mechanisms, and there is a lot of papers that suggest these single molecule mechanisms. BDNF comes up again and again, and this is IGF-1 is an example, VEGF, and so on, and of course also at the neurotransmitter level. But at the end, the question is whether that's really a plausible explanation for the complexity of the phenotype. So I would think that these factors are indeed important, they are involved, but they might be already an epiphenomenon. And of course, if you manipulate them, you get an effect, because if you take out noradrenaline or BDNF, you will have an effect because they are involved, and they might be even importantly involved. And because it's a network situation, if you manipulate the network by one factor, you disturb the network such that you don't get the original regulation anymore. But that doesn't mean that the network doesn't exist, that it's really a monocausal relationship. So from all we know, at the end, it's a very complex regulation, and many pathways can lead to Rome. Many perhaps also varying pathways can lead to these results. When you look in the historical literature, there's many has been a lot of discussion on how enriched environment works. Is this an arousal phenomenon just by general activation? Is this something to do with learning? Is this and that then at the end was what seems to be the strongest hypothesis, that it has to do with development. And if we look at development, then of course we have many factors playing a role. And I would actually favor this idea that it's really development, and hence quite complicated or complex.
Okay.
Thank you.
Anybody else? Please also go ahead and unmute yourself if I don't see you. Gerd, possibly in the meantime, so of course it's possible that there's some kind of random initial event that then kicks off kind of self-reinforcing dynamic leading to individual variability. On the other hand, mice obviously also have a social life and social hierarchy. So to what extent do you think this kind of social parameters are setting more or less the bedrock in which this individual variability may be constrained? And if so, do you see any gender effects?
Gender effects we don't know yet, because we have studied only females, but this is clearly on the agenda, and it would be very interesting to look at. With respect to the social component, I think this is clearly important. So the question of social factors as driver of enriched environment effects has always been strong. So one usually thinks of social, motor, sensory, and more cognitive processes as drivers of these effects. And social, they're of course not completely independent, but these are the classical four categories. And social mechanisms are clearly strong. We have done one study many years ago by observing animals in such enriched environments, and also look at the emergence of some social, some trajectories in social behavior. This is very difficult to do with that many animals in such a large cage. But again, there was some indication of such stable trajectories and behaviors that become more variable with time. And it seemed in that study, it has to be taken with a grain of salt because of the difficulties of doing this kind of analysis, that the animals that are sort of the more active, the more explorative animals in the sense of these trajectories, as I shown them, were also the ones that were a bit more, if you could say, individualistic. They were not antisocial, they were socializing where the others were, but they were not the ones that was always in the middle of the cuddle, and that were perhaps a little less playful than others. Clearly they spent more time exploring their worlds. So this is a very first and very small indication that there might be such trajectories of social behaviors that might correlate with what we see here. And I personally think that this is a very important factor that we are looking at.
Great, thank you. We have another question by Rory Boyle. So please activate your camera.
Oh, I'm very sorry. I think I pressed unmute by accident, so I'm just listening along. I don't have any specific questions. Sorry about that.
Oh, okay.
Sorry.
This is Allison. I have a question.
Yes, please. Hi, Alison.
Hi. So there's this phenomenon in amnestics where they tend to re-explore the same areas, and there isn't this habituation that you see that because they don't remember where they've looked. And I was wondering if you've thought about looking at that perhaps in animals that may have hippocampal degeneration. It'd be really interesting to look at repetitive exploration.
That's a very, no, I've never thought about that, but it's an interesting question. But it goes a bit into the direction of the question we had before, but what is causal here at the level of the hippocampus? And of course you could extend that, you can easily look beyond neurogenesis, which we would primarily do, and ask for general mechanisms of learning and memory, and including the effect of, if you don't have good memory, what happens to your trajectories. Yeah, very plausible idea. I like that, but
or at least look at consistency in exploration.
Yes.
Some perceived learning. Yeah.
Yeah, you can, of course, you can look at the behavior trajectories, and that's the first
Exactly.
pass. Yeah, that's a great idea. Yeah, one can, once you start thinking about this, there's plenty of ideas.
Lots of great data to play
only
with.
so
Yes.
much time and money usually to do it. But this is actually very, very close. When I'm sure that once we have these data sets, one can look at, or more of these data sets, one can look at that even in the absence of a clear degeneration model. And if you use a degeneration model, then of course it becomes even more obvious. Yeah, good. Keep that in mind. Thank you.